So there are some structures against which you design the drugs, right? That a chemistry person is required for synthesizing the molecules. So I also know a little bit of chemistry. That's why I am in chemistry department. So understanding more about the structures against which we can design the drugs, right? So I will be starting with, this is the title of thesis, that is design of therapeutic peptides using structure-based drug design approach, right? So that let us keep it interactive. Teacher की आदत है बीच बीच में teacher encourages asking questions and replying to that, right? To be more specific about the concepts that you should grab out of this 1 hour presentation. So I'll start asking a question. Do we discover the drugs or we can also design the drugs? What is a drug actually? What is the purpose of taking a drug and what a drug molecule does in your body? Anybody who can answer? Please. Your good name?
Shreya.
Shreya. You are from?
I'm from
Tipsuru. Good.
So basically, your molecules are intake for any disease, part or any to cure any disease.
To cure a disease. Diagnosis also. Those are diagnostic molecules. Right.
And also for prevent any further expanding of.
So anything that cures an ailment happens to your body will be called a drug. To generalize the statement, anything that cures an ailment that happens to your body will be called a drug, right? Then what this drug molecule will do to cure that ailment is the question. To be more specific, this is the question, that what the drug molecule will do so that the ailment gets cured. The what? Answer to that, if anybody knows, has gone through in any of your syllabus curriculums, that drug molecule does this thing and cures the ailment which happens to your body. Your good name first and your Sonu. Sorudeep. Tejpur University. Enzymes. Right? Some enzymes are continuously working and doing the housekeeping functions of your body, right? Anything wrong happens to those housekeeping functions, then you get the ailment. That is one thing, right? And to cure that ailment, that protein has to be regularized. If it is in excess, that has to be inhibited, concentration has to go down, then it will function properly. If it is less, is not doing the function properly, it is given from outside, right? So these are the target proteins which are deficient, either deficient in the body or in excess. They are expressed in lower concentration or higher concentration that you diagnose from your blood test. One particular protein is less, then there is problem. You want to express it more using the drugs, right? Other thing is, when some pathogen has attacked, then what happens? Some external pathogen has attacked. That happens inside. Your body functions are not properly running, then that happens, right? What you told. But when an external pathogen attacks the body system, then what happens? She belongs to my lab and she is coming at the end. So, then what happens? Yes. Weighing a person, which is not even visible by your naked eye. Then what the pathogen will do? No, only two are interacting, rest others are not interacting. I haven't heard about these things earlier, right? But the best part a chemist can do with your academic curriculum is something you should approach going into drug discovery process, because there are very less people in that segment and we desperately need new drugs in the future. Otherwise, things are going to be very bad. One virus came, we were helpless, we couldn't do anything faster, right? So many permissions are required, that process was eased out, and somehow we passed through that phase safely. So many lives were lost. So these people who are sitting here, if some of them get interested in building a career, research career in drug discovery process, that will really help our society, right? So that is my purpose, to make you interested in that drug discovery process, right? So let us continue with the presentation. Another question: do we really need to synthesize new molecules? We already have millions of molecules available with us, which are either synthesized by the chemists throughout the world, or were isolated from the natural products like plants and sea products and other materials, some animal species, some bacterial species. So many natural products are isolated and many of them were synthesized. So millions of molecules are already available. You never tested those molecules. Very few were tested for whether they have a potential to work as a drug or not. But most of them are not tested yet. So that database can be used for discovering a new drug out of those molecules also. So we can build the databases of small molecules and use them against the targets, drug targets, and we can work with them. Fine. Next is, the process begins with search of new drugs, then how to plan a strategy, either synthesize new molecules. Earlier people were doing that way only. They were synthesizing thousands of molecules and adding those molecules to the pathogen and trying to see whether any of these molecules is able to kill the pathogen or not. If the pathogen is killed by some molecule, that would be tested on animal. Animal is also killed along with the pathogen. That the molecule is not a drug, it's a poison. So poison will kill everyone, right? So another borderline which comes out is the difference between a drug, a poisonous substance, and moderately in between the two, that is a toxic substance. What is the difference between the three? A poisonous substance, a toxic substance, and a drug molecule. You have heard all these three terms, no? So what is the difference between the three? Anshika. So we are keeping it interactive so that everyone understands the concept properly, right? I am not going deeply into my research, what I am doing. I am trying to make you understand the concept so that everyone can start working on this, taking whichever pathogen, whichever ailment you want to work on. You can start with that and you can do your own things, right? That if you understand this concept, right? So what was the question? Toxic substance, poison, and drug molecule. Have you heard that some of the names which are there in homeopathic medicines, right? Snake poisons, crate venom, cobra venom, some natural products isolated from cobra venom, diluted to an extent that you will see hardly there is any molecule. इतना dilute कर दिया उसको कि molecule है भी के केवल alcohol के drops पिला रहा है, ऐसा सोचते हैं. आर्सेनिक दे रहा है उसमें, आर्सेनिक तो हम सुनते आए हैं पॉइजन है, so they are adding arsenic, but in such a low concentration that it may cure a disease. A poison may also cure a disease, right? You must have heard that people are worshipping on the hills of mountains and snakes will also bite, right? Other species will also bite, which are secreting deadly venoms. They are also biting them, but nothing happens to them. They are not even affected by the temperature. Such a low and high temperatures, they remain unaffected. How they build such a strong immune system, which is helping them getting rid of all those types of problems. Right? Snake is biting, injecting milligram quantities of venoms, which are deadly even in microgram quantity. If a healthy person living in a city consumes that, that will kill the person. But milligram quantity is not killing the person worshipping there. How? Even the soldiers get trained to snake bites and other things. How that happens? Those venoms contain some poisonous substances, which will bind to some of the proteins in your body, which are housekeeping proteins. What are housekeeping proteins? Name any housekeeping protein in your body. The most common one, doing some housekeeping functions in your body. Hemoglobin. What does it do? It transports oxygen from lungs to all the tissues, all the cells of your body. And the byproduct, which is carbon dioxide, it takes it back to the lungs and allows it to exhale from the body. Right? So it is the best function of a molecule, a protein, which is doing in your body and helping you breathe out and oxygenate all your tissues and cells. Right? So if if some molecule disables that protein so that hemoglobin is not able to function, the function which it is doing is not able to perform that function, then you will die. Right? Similarly, there are some neuroproteins, there are so many receptors which are there on the surface of cells. Some factors, proteins bind to them, some hormones bind to them and transduct a signal inside the cell so that the machinery activates and synthesizes more and more housekeeping proteins, and they come out to your bloodstream and they perform their own functions. So any of those housekeeping proteins, if get inhibited by some molecule, then you are in trouble. Right? And for your survival, you are synthesizing, your system is synthesizing hemoglobin and 20,000 such type of proteins, which are doing some housekeeping function, one or other, in your body and helping you sustain your life. Right? Anything goes, level of any protein goes down, then you are in problem. Any level of any protein goes up, then also you are in problem. Excessively working enzymes and deficiently working enzymes, both are problematic. So one thing is that. Other thing is, a pathogen which attacks your body injects its genetic material into the host cell. Host are you are the host for the pathogen. It will utilize your own cells and will inject its genetic material, will express the proteins. Like we express housekeeping proteins, these pathogens also express their proteins. Using them, these proteins, they replicate themselves, they multiply themselves, and when a pathogen which is attacked, single virus or single bacteria has attacked and is multiplied to thousands, multi-thousands and millions in number, then the secondary symptoms in your body starts coming. You start having nausea, you start having fever, you start having pains in your joints and other things. Right? That ailment will start appearing into your body when the number of pathogen has multiplied to a considerable extent. Then you start experiencing that. Right? So we can synthesize the new molecules wherever it is necessary. Otherwise, we can utilize the databases which can be built from the molecules which other scientists have built over the years. And those molecules are not utilized whether they have a drug potential or not. So we can see whether these molecules are also able to cure some of the ailments. like homeopathy medicine is doing. So the difference between a poison, toxic substance and a drug lies in the concentration. Some of the things which are good for your health, basil leaves, you are using, you are adding in your tea, and you get refreshed after having a cup of tea by adding some of the basil leaves. Right, that is a herb which is curing the ailment. तो थोड़ा सा headache है, थोड़ा सा कुछ और problem है, that gets healed when you consume that. But can you consume 100 grams of basil leaves? It becomes toxic. You'll start having problems. You consume more, you may even die. The thing which was acting as a drug becomes toxic in higher concentration, and in further higher concentration it becomes a poison. Fruits, you consume 1 or 2 fruits, you feel better. You consume 10 apples, you'll start having stomach ache. More than that can even kill you. Right? So there is a particular concentration of everything. Even potassium cyanide, which is the deadliest poison, has some concentration that kills you. Microgram or nanogram, whatever. Lesser than that concentration may even cure some of the ailment which has happened to your body, right? That even lesser than that concentration can remain in your body, and you will not having any symptoms, right? So the concentration which is lower or higher for one particular substance can make it a poison, a toxic substance, or a drug molecule. How it started? Ancient systems, when we started discovering the drugs. How it started? Okay? Ancient human beings, they didn't know anything about whatever is created by the nature, right? So they started eating everything. Then they started understanding the difference between eatables and non-eatables. Primarily on the basis of taste. This thing is good in taste, so should be good for us. This thing which is bad in taste should not be used. But later on, the wisdom came, and they started discovering some of the things which are bitter in taste are also healthy for your body system, right? They are also giving some relief from some of the ailments. So they started calling them herbs. The plants which can be consumed in smaller quantity, they can get relieved. They can help you get relieved from the symptoms of some of the ailment that has happened to your body. Some of the extracts can be taken up and applied on your wounds, and that can heal the wounds faster. So those having antibacterial properties, they didn't know about the bacteria that time, but they were having some antibacterial properties, and when they applied, like neem, when they applied, that ailment or the wounds get healed faster, right? So that is how they started discovering out of the eatables and non-eatables, and then the herbs out of the eatable and non-eatable substance. Then later on, the chemical era started, right? Up to this thing, ancient humans, and what they were doing is the plant extracts which we were applying or we were consuming was up to the 16th century. 17th, 18th century, we started discovering or taking out the ingredients of those herbs through chromatographic methods, that this component of the complete herb substance is the one which is active, which is the active substance, which actually is curing the ailment, right? That is the chromatography era. 17th, 18th century, we started isolating the active ingredients from the plants, right? Then the chemical era, after 18th century, we learned to synthesize new molecules, and we started synthesizing those molecules which were part of the natural products, okay? So natural or plant-driven products which were used as such earlier, then we started isolating the molecules out of them which actually were curing the ailment, and then the synthetic era which started, and we started modifying these structures to get better drugs, which were 10 times, 50 times, 100 times more potent than the actual ingredient which was there in the plant. Just simple modification of the structures gave better drugs. But that didn't happen overnight. Thousands of analogs of that molecule were created, and they were applied on mouse models or directly on humans. We don't know what happened, how they discovered them, but the more potent molecules were discovered from the products which were isolated from the plants, simple modifications of the structures, but still they didn't know what this molecule is going and doing into your body system, right? Then later 18th century, biochemical era started, where we started discovering the mechanism of happening of a disease, right? Some pathogen has attacked, what that pathogen is doing into your body, what type of proteins it is expressing for its survival, and which proteins are responsible for the ailment which has happened to your body, right? So rationalizing the complete process, how it started, now we are not starting with the molecule which we isolate from the plants, we start with the disease. This disease has happened to your body. Why this disease has happened? Some internal factors, right? Some of the proteins are not expressed because of lifestyle disorders, right? उस समय पर नहीं था, अभी ज़्यादा होने लगे, अभी हम तो आराम करने की ज़्यादा आदत पड़ गई है, राइट? गाड़ी में हम लोग घूम रहे हैं एसी चला के, जो बाहर का जो हीट है, जो 45 का टेम्परेचर है, वो हम एक्सपीरियंस नहीं कर रहे, राइट? सो द प्रॉब्लम स्टार्ट्स विद द लाइफस्टाइल which has started from the generation which is surviving now, right? Earlier, the primary reason was a pathogen which has attacked the body system, that is bacteria, parasite, virus, fungus, or any other pathogen which has attacked your body system. Then how this pathogen, क्या भाई ये नहीं काम कर रहा है? इसका पॉइंटर काम नहीं कर रहा है भाई. Anybody knows how the pointer will work? Yes. So this is how the process was rationalized. Now we start with the disease, and we now understand the mechanism of the disease. So the process of replication or multiplication of the pathogen in the host or body system, for its multiplication, it will inject its genetic material, that is DNA or RNA, into the host cell, right? And will express all the proteins. That genetic material will express the protein. That is, the process is called transcription and then translation. So translation of all the necessary proteins. Some proteins will make the outer covering of the pathogen, some protein will make the nucleus, some proteins will make the enzymes which are required for its functioning, and many other proteins, right? When the complete mature pathogen will be synthesized, that will come out of the host cell and will infect other cells. That is the multiplication, that is the process of multiplication of the pathogen, right? And the biological mechanism of its multiplication. So what as a chemist and the drug discovery scientist, what we want to do, the process in which we are interested to discover or design a drug, we need these target enzymes which are responsible for survival of this pathogen. Some of the enzymes may be common in your system as well as in the pathogen system. If you inhibit them, you will be in problem. That will give some toxic effect, right? Because your own housekeeping functions will be inhibited. So you better distinguish between the proteins which are expressed by this pathogen, which are not common in the human system and the pathogen system, right? You discover them, you mark them, and then find out the inhibitors of that enzyme, which will inhibit the function of that protein which is responsible for the survival of pathogen. Then the pathogen will be in trouble, right? For prolonged time, if you stop the function of some of the housekeeping enzymes of the pathogen, then pathogen will die, will not be able to multiply. That is the purpose, right? So next is, now we understand the disease. We have selected some target macromolecules, not small molecules. These are the proteins or macromolecules inside the pathogen system which are governing, which are regulating some of the housekeeping functions of the pathogen, right? We have selected any of these target macromolecules. Usually they are enzymes, right, doing some housekeeping function in the pathogen, and discover the structure, which is very important thing, right? These structures, so many biochemists and scientists, they have, whoever is working on that pathogen and the proteins expressed by that pathogen, they will isolate, they will clone that protein, they will try to crystallize that, and finally they will discover the structure. Whoever discovers the structure first will add the coordinates on the protein data bank, that is PDB. RCSB protein data bank. You have thousands of structures available on the PDB where you can utilize that database, and you can isolate the coordinates on your computer screen and visualize the structure, and you can also discover how that protein functions, which are the substrates it is catalyzing, right? So substrate is the primary informative material to discover your molecule of choice, right? Then you discover the ligands which will fit into this structure and will inhibit the function of that protein. Getting it? Any questions you can ask in between. Whenever you are not able to understand the concept, you can ask anytime. So to understand, those who haven't gone through this, an enzyme binds with a substrate into the active site. The whole enzyme or the protein is not the active protein. Only some part of the protein will bind to the substrate, will make some changes into the substrate, and will synthesize the products for which the enzyme is able to utilize that function and will bind with the substrate. What you are doing is, so this substrate will bind to the active site of the enzyme, right? Whether breaking of the bonds is required or joining of the bonds of small molecules are required, those any type of function this protein is doing and is utilizing the substrate molecules and furnishing the products. What you are going to do is, this is the substrate which is binding to the protein and making some products. The enzyme is functioning properly, right? Your molecule, what you are going to design to inhibit that protein function is a similar molecule, similar in structure with the substrate, which will go and bind to the active site of this enzyme and will not come out, right? That will bind more strongly than the substrate. This will compete for the active site from the substrate, will not allow the substrate to bind here, and will completely inhibit the function of this enzyme. This enzyme is specific for the pathogen, is doing the housekeeping functions of the pathogen. If this protein gets inhibited, then part or the cascade of reactions of the pathogen will be disrupted. Pathogen will not able to multiply. आपका hemoglobin को किसी ने inhibit कर दिया, you will die, you will not be able to survive. So any of the housekeeping enzyme of the pathogen, if you inhibit with your molecule of choice that is similar in size and structure of the substrate, but will bind more strongly to the active site and will not allow the substrate to bind, then you have discovered a molecule which can become a drug. Is not a drug. This can become a drug. Okay. How fast these enzymes function? आप कहते हैं कि lightning fast speed से काम कर रहा है. किसी का जो high speed से काम करने वाले को you usually say that it works with a lightning fast speed. enzymes even work with much more speed than the light, right? So this is the normal function which one of the protein in your body, that is called catalase, is catalyzing, is every time there is oxidation in the body system and this molecule, small molecule, that is hydrogen peroxide, gets generated. This needs to be eliminated very fast from the body system. If this remains, it will oxidize everything, right? If this gets in higher concentration, this will oxidize everything and radicals will be generated, right? So body has devised a mechanism to eliminate this very fast. This is the normal thing. It will degrade into water and oxygen, into the molecules of water and oxygen. This is the normal degradation of hydrogen peroxide, which doesn't happen by itself. If you add ferric ions to this, the rate of reaction, rate of degradation becomes 30,000 folds more than the normal degradation process, right? And that enzyme which is there in the body to degrade that faster is called catalase. If you add this, the speed becomes, count the number of zeros, 100 million folds, right? Light की speed कितनी होती उससे कहीं ज़्यादा है, right? And where is the protein and where is the active site? This you can see here is the protein, structure of protein, and this small part is the active site where H2O2 will bind and will release water and oxygen as products. It is a housekeeping enzyme in your body which is eliminating the toxic substance which is called hydrogen peroxide. So these enzymes can do both the functions. They can join some small molecules and create a bigger product, joining of the bonds, and also they can bind a bigger molecule and break the bonds between the bigger molecule and generate smaller molecules, right? And what are the things for which these proteins are made up of? Amino acids. So amino acids, 20 types of amino acids, they join in whatever permutation combination which are required for that protein to function, right? Function of the protein is dependent on the structure, and the structure is dependent on the composition of amino acid, the sequence of amino acids in the structure, right? So these amino acids will align, will join together and will make the primary, secondary, tertiary, and finally either the tertiary structure is functional or some subunits join together to make the quaternary structure. This you all must have studied in your classes, right? So amino acid chain which is joined in whatever the composition this protein is, they will join and they will make the primary structure, which is the primary chain of amino acids joined by peptide bonds, amide bonds usually, right? And there are some secondary and super secondary structures which are primarily made up of regular repeated patterns of hydrogen bonding. Two patterns are there, one is beta sheets, other is alpha helices. Alpha helices is intra-chain, within the same chain, continuous hydrogen bonding, first to fifth, second to seventh, sixth, third to seventh, fourth to eighth, like that. So continuous hydrogen bonding is there within the chain, and the helix will keep on generating up to the length it can generate by itself, right? Similarly, the beta sheet is intra-chain, two parallel or anti-parallel chains join together and make crossed hydrogen bonds between them, and up to the length this chain continues. Those structures are found in parts in a complete protein structure. Somewhere beta sheet is there, somewhere helix is there, somewhere random chain is there. But the secondary structure is made up of regular patterns of hydrogen bonding. Third type of thing is when finally the structure folds and joins and gets compacted because of all the supramolecular interactions, which are hydrogen bonds, which are hydrophobic interactions, which are stacking interactions, and which are other interactions, or all sort of interactions. They will join together and make the complete functional proteins. Most of the proteins has only tertiary structures, right? But some of the proteins like your hemoglobin has quaternary structure. Two different subunits formed by two different gene products, right? So sequences are different, alpha 1, alpha 2, beta 1, beta 2. So it's a tetramer, and all the units are not same. It is alpha and beta, so it will be called heterotetramer. Some of the protein having two subunits, one different from other, then it will be heterodimer. Some protein is having four subunits, having all the same type of subunits, that will be called homotetramer. All subunits are same and joined together. So four subunits are there, homotetramer, two subunits are there, homodimer. Fine. So this is how it looks like from a software which is called PyMol. The protein active site is there, some cavity in the complete protein structure where a ligand is going and binding, right? So this will be called binding site or binding pocket, where a small molecule is going and binding. You can see in a complete protein structure, there is an active site and there is one small molecule which is there inside the active site, is making some interactions with the protein, right? Some of the residues of the protein, it is making some interaction, which are usually responsible, this threonine 106 and methionine 109 and other part of the protein structure in the active site is binding with this, and sometimes they are also responsible for making chemical change into the substrate structure. How important this three-dimensional structure is? When you draw a molecule, it looks like this, right? This is the peptide synthesized in our lab. So this looks like this if you draw it with a pen and paper, right? But when you actually crystallize this and it folds and it gives a three-dimensional structure like this, where some hydrogen bonds are formed, some other interactions are formed, that will be visible only when you make a three-dimensional structure of the molecule. So this 3D structure can be obtained by so many ways, right? The best one is crystallographic methods. You crystallize the protein, do the X-ray crystallography, single protein or single molecular X-ray crystallography, right? Small molecule can have a different setup, and proteins have a different setup, right? You can determine the structures by X-ray crystallography of proteins and small molecules both, right? So single crystal X-ray crystallography for small molecules and protein crystallography for protein molecules. If you are not able to crystallize the protein, then you can utilize NMR structures of the protein, right? Larger quantity is required to get the structure from the NMR. Other thing is computational methods. Nothing is available, even the protein source is not available, but protein is there, you know the sequence. Then you can model the protein utilizing the structural parts of that protein which are there in other proteins, right? There is a software discovered 2 years ago that is called AlphaFold. Heard of it? The one who discovered got the Nobel Prize. So that changed the scenario. All the proteins which are not available, you have the sequence of the protein, you add the sequence, you will get the 3D structure. And those structures which were discovered later were very similar to the one workable model which was discovered by that method. So computational AI/ML methods you can utilize, and you can find out the structure for which the protein is not available. Even those structures are discovered, right? Now you start working with that. To be more precise, how actually we are going to discover the drug molecule. That was the process by which you utilize to select the target macromolecule. Now you know the target macromolecule, and now you are searching or synthesizing a new drug molecule. So the process starts with the disease, then biochemist will tell you the mechanism of the disease, will tell you the pathway, enzymes or proteins which are responsible for survival of that pathogen, and then you discover some of the protein or target macromolecule which you can easily work in your lab, for which the structure is available. Best is the structure is available with some bound ligand already which is there inside the active site of the protein. Right? If that is there, that is the best thing. Now you also know the active site. If you have the native protein structure not bound to anything, then you will have to take extra effort to discover where is the active site, where you want to bind your molecule so that it is not able to do its normal functioning. Right? So you need to block the actual active site of the protein. So you discover the 3D structures in the RCSB PDB protein data bank, where you will get the structure with bound ligand into the active site. You remove that and you start talking your molecules from the database, right? Then you get the information about the protein and the active site. If you have the structure, you also know where is the active site, which are the amino acid residues inside the protein cavity, active site, which are responsible for catalyzing the function, right? You want your molecule to replace the substrate and bind there with a stronger affinity, stronger binding affinity. Fine. Now you start hunting for the ligand. You can generate your own databases. You can utilize the databases which are already there. One is zinc, ZINC, zinc database for small molecules. Other is already discovered drug molecules, drug data bank. FDA approved drugs, 2700 drugs which are which patients are consuming, which are there in the market, are FDA approved drugs. Their data bank is available. 26 or 2700 drug molecules for different purposes, for malaria, for cancer, for diabetes, for this and that. 2600 drug molecules FDA approved are there. You can utilize that data bank also. The drug which is working on malaria can also work on coronavirus, right? You know this? Donald Trump was president that time also, in the corona era, right? And he threatened Modi, you give me hydroxychloroquine, otherwise I will send missiles. Do you know that? Why? Why hydroxychloroquine was so important that time? Hydroxychloroquine, eventually, when all these processes happened, we started discovering through the drug data bank, and we finally got that these molecules can work on coronavirus to control that. So this is called prophylactic molecule. If it is already there in your body, in your blood serum, then virus will not affect you. Virus has already attacked, then you take it, then it won't cure that. But the virus attacks when this molecule is already there in the body, then virus cannot multiply. Those were called prophylactic molecules. So some of the molecules which work on malaria, they were also acting as prophylactic molecules. So larger quantity, because we are getting affected, America is not getting affected with malaria. They are not even bothering, and they are not working on malaria disease. We are struggling day and night to find more molecules against malaria, right? So that we have piled up a huge stock of hydroxychloroquine over the years, and we were safe because that was distributed to our housekeeping staff and sanitation staff and everyone who were working outside and helping the patients which are there. So that molecule was required, and they threatened at that time that if you do not give this, then we'll do that. So this process of virtual screening helped us during that time. We discovered some of the molecules. आपको ध्यान होगा कि उस समय में किसी को अगर घर में अगर किसी को अगर हुआ होगा, then the first line therapy which was advised to everyone was ivermectin, doxycycline, and hydroxychloroquine. This hydroxychloroquine is also consumed not even in malaria, is also consumed by arthritis patients. The one having arthritis continuously uses it every day, right? तो जिनको arthritis था और जो hydroxychloroquine consume कर रहे थे, उनको नहीं हुआ. Even with multiple disorders in their body, they survived coronavirus because they were already consuming that molecule. right? So these three molecules were discovered by this process, and this process is known as repurposing of the drugs. Note down, the repurposing of drug molecules. As the term specifies, what should be the repurposing? What is the purpose of repurposing of drugs? Your good name? आस्था. repurposing से क्या समझ में आता? मलेरिया के लिए यूज कर रहे हैं, इसमें अप्लाई करके देखते हैं, क्योंकि ह्यूमन ट्रायल्स हो चुके हैं, सेफ है, इफेक्टिव है, ज्यादा भी लिया जा सकता है, बिना जरूरत के भी लिया जा सकता है, मरेगा नहीं कोई खाएगा तो, ठीक है, और सेफ है, जो सारे फार्माकोलॉजिकल पैरामीटर्स होने चाहिए वो सब उसमें है. तो आपको ड्रग ट्रायल नहीं करने पड़ेंगे, आपको केवल इसका दूसरा वाला जो इंडिकेशन है उसके ट्रायल करने, उसमें काम कर रहा है या नहीं कर रहा. इफेक्टिविटी ट्रायल्स आपको करने हैं, आपको अदर ट्रायल्स नहीं करने हैं. बाकी फेज 1, 2, 3, 4 वहां तक नहीं जाना है, डायरेक्टली फेज 3 पर चले जाइए और देखिए कि इफेक्टिव है या नहीं है. सो दैट इज अ गुड थिंग. इफ यू यूटिलाइज द डेटाबेस ऑफ FDA approved drugs, you can discover a new drug very fast, because just you are repurposing the drug against the new ailment which has came out like that. Okay. So hunt for these small molecules either through the databases or create your own database, and then what you will do is, with the help of software, you will replace the molecule which is already there in the active site for the PDB which you have selected. You remove that molecule with the help of software and dock your own molecules in the database into the active site. Right? One by one, the software will dock everything and will give you the scores which are giving better affinity than the. अब इसमें सबस्ट्रेट भी लगाइएगा, या जो ऑलरेडी अवेलेबल इनहिबिटर है उसको भी साथ में डॉक कीजिएगा. If your score, your binding affinity is better than the one which is already there, then you are in business. Okay. And then the hits which are generated, that has not become a drug. You have found a better inhibitor than the available inhibitors. Now you optimize by doing actual activity. Devise a system for doing the actual activity for those lead molecules which you selected out of the docking, and then do the medicinal chemistry, modifying their structures. You have got the scaffold. That is the best thing, right? Now you modify. कहीं OH group लगाया, कहीं NH लगाया, कहीं पर आपने amide bond लगाया, कहीं aldehyde लगाया, whatever best suits your structure and still binds into the active site. You play with the structure, play with the functional groups, and then finally you can select, you can discover a new drug molecule. Activity has to go parallel with the new designed molecule. Now you can start synthesizing. अभी तक synthesis की जरूरत नहीं थी. Earlier process was thousands of molecules unnecessarily were synthesized, and then they were tested directly on the pathogen without knowing where it is going and binding, what it is doing, right? That process we started discovering later on. Now we know the process, we know the target macromolecule, we know the functionality, and we are going to block them specifically with our own designed molecules. First hunting those molecules, better into the active site, and then optimizing them for activity to get the druggability or drug likeliness of the molecules. There are software to calculate drug likeliness of the molecules. Anybody who knows that? Lipinski, ADME, right? ADME, that is absorption, distribution, metabolism, excretion, toxicity also, right? Lipinski's rule of 5 helps you find out whether your molecule is going to work later on or it will be filtered out in the process for drug development. So many things you can do. What we are doing, one example we'll take from the work that my students are doing. Malaria, you know, is a problem which affects Africans and Indians. Europeans and Americans remain unaffected, so they are not bothered. So we are the ones who want to discover new drug every time, because the earlier drugs get resistant and they are not working on the pathogen. Pathogen is very smart. Pathogen keeps on mutating its 3D structures of the proteins against which you are trying to discover new drugs. So some of the residues in the active site, they will change. Now your molecule which was binding with a better potency earlier is not able to bind. So either you continuously keep on discovering new drugs or find better versions or whatever, new scaffolds through docking and these experiments. Earlier remedy, cinchona bark, प्लांट एक्सट्रैक्ट वाज कंज्यूम्ड, ठीक है। आज आपको पता चला है कि मॉस्किटो बाइट से आपका जो प्लाज्मोडियम पैरासाइट है ये बॉडी में जाता है और वो ऐसा ऐसा करके प्रॉब्लम रहती है, राइट। दैट अफेक्ट्स द रेड ब्लड सेल्स एंड दैट किल्स all the red blood cells and you get problem because of malaria, right? Later on that happened, that used to continue till 1632 when the plant extract from cinchona bark, right, was utilized. Later on, after 200 years, just around 200 years, we discovered that this is the molecule responsible. This molecule is there in the cinchona bark, which is actually curing, helping to eradicate malaria. And then just around 200 years later, there is no going back. If you go ahead, then you'll have to go back to this. Fine. 100 or 125 years more were used for synthesizing, to be able to synthesize this complex natural product, right, which is called quinine, that was extracted from the cinchona bark, right. This is the actual chemical era that started in mid 1900, where we actually started synthesizing very complex organic molecules. Right now you can synthesize anything, then you can bet upon whatever is you are taking out from the plant extract, you start synthesizing them. Then medicinal chemistry, you start modifying the structures to get better molecules, right. After that, what happened? This molecule was so effective that when a pandemic came, just like corona pandemic came, malaria pandemic came, then they started adding this to your flour and your salt. Everyone starts consuming to get rid of this pathogen, right. ये इतना ज़्यादा कंज्यूम करेंगे, then what happens? Gradually the resistance will come, okay, and this molecule became ineffective and was taken out from the market in 2006 when WHO declared this as ineffective or resistant molecule against the parasite, and the use was discontinued in 2006. So the best molecule which was there, trying to eradicate the parasite, got eradicated itself in 2006. We had new molecules by then, so we survived somehow. This is how this small creature survives in your body system. Mosquito bites here, it injects sporozoites into the system. First, these sporozoites go to the hepatocytes in your liver. They affect the liver cells. There some of the stages happen and they multiply themselves, they get matured, and in the form of merozoites, they come out of the liver cells, hepatocytes, and then they enter the bloodstream where they infect red blood cells, right? They get further matured in the erythrocytes, and in the form of gametocytes, they come out into the blood, right? And from your blood, a new female mosquito, Anopheles, takes it out, and some of the process happens inside the mosquito body, and finally it creates more sporozoites, and sporozoites are injected again into the new person who gets the malaria parasite. Right? This is the process, and this process, how you are going to work with this process, take a suitable enzyme target from the malaria parasite. ठीक है, अब ये स्पोरोजोइट्स में हो सकता है, ये मेरोजोइट्स में हो सकता है, और ये मॉस्किटो बॉडी में जो प्रोसेस हो रहा है उसकी हमें ज़रूरत नहीं है। मच्छर के अंदर हो रहा है, वहाँ पर मारोगे तो उससे अच्छा मच्छर को मार दो, वो ठीक है। आपका लीवर में जो हो रहा है और आपका ब्लड में जो हो रहा है उसको आपको रोकना है। So there are so many targets in the liver and so many targets in the bloodstream where you can stop any of these processes to get the mature form of this parasite. Right, and these are so many processes which happen. There are peptidases, there are ribosomal factors, coenzymes, amino-acyl RNA proteases, and some of the enzymes will help synthesizing the fatty acids in the body of parasite. Some of them will synthesize the proteins. Some of them will cleave the proteins and will generate new amino acids. So many pathways they catalyze. Any of these enzymes, just around 42 target enzymes are available. Okay? Very few of them are common in human and parasite, but you can target just around 30 out of these 42, and you can work with any of the enzyme. Okay? You can also select multiple targets. One target from hepatocytes, one target from एरिथ्रोसाइट्स. ठीक है, दोनों का मल्टी टारगेटिंग हो गया आपका, वो भी किल कर रहे हैं, इसको भी कर. बेस्ट इज टू जनरेट अ मॉलिक्यूल विच इनहिबिट्स वन ऑफ द साइकिल ऑफ योर हेपेटोसाइट एंड अनदर साइकिल ऑफ योर. ठीक है, दैट इज अ वंडर ड्रग if that is inhibiting two targets, one from the liver stage and one from the blood stage. 21 are listed here. Actually there are 42, right? So what we have started working is taking out two molecules, two macromolecules. One is DHFR. DHFR is common in human and parasite both. Why we selected? Because the already validated target is this. Many of the drugs which are already there in the market are designed against these two. So it's not completely inhibiting, it may be taken as a regulating overexpressed enzyme. If for some time, if you inhibit in your body also, that will not, your system will not die. Okay? Some of the protein will keep on expressing, and for some time if that process is stopped, then there is no problem. Primary goal is to stop the parasite from multiplying. So this was taken as the one of the target because people were already working on that. The new target which was selected was PFENR, that catalyzes the fatty acid 2 pathway of the parasite, which is important for its survival and is not common in human and parasite. It is only there in the parasite. Right? Other two targets which now my students are working is plasmapsins, that helps in the heme degradation, for which you get anemia and this jaundice, and that later on after the malaria is over, then you start experiencing jaundice because red blood cells got degraded. So this is degrading the hemoglobin in your system, that is plasmapsin 2. And one more target is purine nucleotide phosphorinase, that is purine salvage, right? That is one of the factor which is responsible for your synthesis of nucleic acids, right? So protecting that is for the survival of pathogen is also one of the targets. So fatty acid 2 pathway is there only in the pathogen, is not there in human, so it's a good target. That is for PFENR, right? So one of the structure which was bound with the inhibitor which was already there, that is triclosan, which is used as a common antibiotic in the soaps, right? But that cannot be a drug molecule because so many chlorines are there in the structure, right? So I started with that in the beginning of my career. I worked as a scientist at CDRI for 1 year. There, the database of compounds which were synthesized by CDRI scientists over the last 20-30 years, that time 2005-6, right? उसके पहले 30 years से जो ये लोग बनाते आ रहे थे, उसका हम लोगों ने क्या किया कि we started drawing these structures and converting them from 2D to 3D. So that database was created. We got exhausted. 30,000 molecules are there. In 1 month we were able to create a database of 10,000 molecules. और लगा कि भाई और ये बनाते रहेंगे तो बोर हो गए, स्टूडेंट भी भागने लग गए. उसके बाद में क्या 10,000 को लेके इनसे काम करते हैं, क्या पता इसमें कुछ निकला, right? So we started working with that database of 10,000 molecules that in-house, that was generated in-house at CDRI, right? So that compound database systems were not good enough to work with all those 10,000 molecules in one go. Then there is another method. You can search for the scaffold which is sitting better into the active site. Search for that scaffold into the database of small molecules. Rather than docking all the 10,000 molecules randomly, you first select the molecules out of those 10,000 database which will be able to bind there and which will not be able to bind there, right? So that you get a focused database. When you create a scaffold and you search into the database through that scaffold, then you get smaller number of molecules. 10,000 से 800 रह गया. उसके बाद में even in 8,800 molecules there were very big molecules which I know will not be able to fit into the active site. Those were selected and filtered manually. 90 molecules were selected and those 90 were docked into the active site of PFENR, removing triclosan out of it, right? So this is how it happened. This is triclosan, which was used as inhibitor because it was not a drug molecule but can be used as inhibitor because it was sitting into the active site. The one who discovered the structure of PFENR also discovered this bound already into the active site. We created this scaffold from triclosan. This was the structure of triclosan. We removed all the chloro. This OH group which is there at this position is helping in binding into the active site. So we cannot remove this. So let us keep this. Let us keep these two ring structures. Let us keep this distance intact, right? And we generated this scaffold, keeping some distances fixed, two ring residues safe and the OH group intact. Search for this scaffold through the database of 10,000 molecules. Right, those 10,000 molecule database created in-house, the scaffold was present in 800 molecules. Then 800 molecules were opened and selected and filtered manually to see which structure should bind to the active site, because there is small space into the active site. A very big molecule having this scaffold cannot bind there. So selected manually 90 compounds, and now these 90 compounds were docked into the active site of the PFENR, and finally 6 compounds with best energy scores were selected. These were the 6 compounds, right? Triclosan was having 2 ring structures. Here we got naphtha ring and 1 hexa ring, right? So these 2 molecules which were there, all 6 should have been there in the database, but only these 2 were available, right? And the molecule which is the sample which is kept there since last 25 years, you can understand what would have happened to that. Okay, that got charred. So we synthesized this molecule, right? This was available, so we purified this, taking out from the database, from the library of molecules which were submitted by the scientists. We purified this one. These 2 were tested against the Plasmodium parasite. So you need to develop the assay, which was already there because CDRI has given a task to work on malaria. So anybody who joins there has to work on malaria, has to keep one project as malaria, and other things you can do of your own, whatever you wish to do. So that system for testing in vitro activity was there, right? So these 2 molecules were tested, and one of the molecule gave very good activity, which usually you will get after synthesizing 10,000 molecules yourself. You will get this activity. Now you got it without making a single molecule. Right, ek banaya, sorry. Right. So one molecule gave this activity, which is the very good initial activity. Now you can optimize this activity. You modify, you play with the structure, keep the scaffold intact, play with the functionality, and then you synthesize. इसमें आपको आसान और क्या हो गया? I am a peptide chemist, so I got to synthesize the amide bond. I got a task to synthesize amide bond. तो पेप्टाइड वाले के लिए आसान है भाई, amide bond बनाने की तो आदत है. So what you need to do here is you take some of the acids, carboxylic acids, and some of the aniline analogs, and you keep on joining that, and you start synthesizing new analogs of it. So whatever acids and whatever amines, aromatic amines which were available, I joined them. I synthesized around 20-25 molecules. Activity got better from 10 to 2, right? And then finally up to 0.5. Just simple modification of the structure optimize the activity to a greater extent, 20 times more than the one which we got initially. If I could have reached up to 0.1, that molecule would have gone to trials. Okay. So that work ended here, and we published a paper out of this, and this was the structure. The blue one is triclosan, and the other one is the molecule that we synthesized, which is better active than the triclosan, right? So that molecule is entering into the deeper cavity of the active site and is binding with a better potency. These are the structures, triclosan compared with the synthesized molecules, and these are the binding scores, right? Here the binding score is very less. That means it is not specific for the active site of parasite. It is doing some other purpose also. It may be rupturing the cell membrane, it may be doing some other thing, but is not giving activity because it is binding there in that enzyme, because the score, binding score is very less. While I have got much better scores with my designed molecules. See, the activity is 2, then it is 8. Activity is more than 2, then it is lesser. Activity is better, then the score is also better. So docking score, what you are getting out of the virtual experiment, is also reflecting into the actual activity if you are doing the experiments carefully, right? Is the perfect example. If you start searching from the scaffold through the database, and then you start optimizing the activity, you will get much better result, and within 1 month of time. 1 महीना डेटाबेस बनाने में लगा, नेक्स्ट मंथ से हम लोगों ने मॉलिक्यूल बनाना स्टार्ट किया. 1 महीने में 4-5 बनाते थे. 6 months work, you got activity which people used to get in 5 years. Within 2-3 months you got that activity, right? So fast this process, these computational methods, if you learn them. In the corona time, people were locked in their homes and they published hundreds of papers using this technique. Free softwares, right? Free databases. Take out the protein from PDB, take out the structures from databases, dock them. If you get some activity, publish them. And that was allowed because experimental labs were closed, so that was allowed. So everyone who were doing some docking experiments, they utilized that era for publishing more and more number of papers. Right, so I am a peptide chemist, so what I will see is the structure which is there, how much length of a peptide I can utilize to work with that active cavity in the housekeeping protein, right? So pathogenic protein taking information from the this, what was that, triclosan. So triclosan experiment and organic molecule synthesis. These dipeptides we can synthesize in our lab frequently, right? So a library of 400 dipeptides was generated in-house. Why 400? There are 20 amino acids. A dipeptide with all combinations mean 20 into 20. 400 dipeptides, virtual library was generated, and we wanted to see whether the dipeptide is also sitting in the active site or not. They are giving much better scores than the earlier molecules, right? These are the scores. These are triclosan, and I asked them to utilize those also. That was giving how much binding score they were giving? Up to 9, 8.9. 8.9, and with the dipeptide we got up to minus 12.8. Right, triclosan is still 9.2 here because different softwares gives you different values. That was the software which I used 20 years ago. This is the new software based on new types of force fields and new calculation methodologies. That is. Sorry? Potential energy. You get better binding affinity if more energy is released, right? more negative the score is, better is the binding affinity. Another example from the same the number of proteins, 42 proteins were there. This is another protein which is working on different target, that is plasmapsin 2 is the protein of Plasmodium falciparum which is responsible for heme degradation, right? That is in the hepatocytes, heme degradation. If that gets inhibited in the first stage, you can tap the virus and parasite, and you can work with that. So this was the one which was already available in the active site, peptide. If peptide is there, then we are on top, because we can modify this peptide and we can generate better peptides than this, right? So what we started doing is we started modifying this sequence, starting with the dipeptide. But this peptide is a heptapeptide, 7 amino acids, and you have a database of dipeptides. Then what to do? How to increase this length up to hepta? Strategically, you can do this. Initially, we docked the 400 dipeptides into the active site, right, one by one, obviously. So we got these docking scores, right? What we observed is there are two dipeptides in the same active site having a space of 5, 6, 7 amino acids, right? One is on this side, other one is on that side, right? One is on left side, other one is on right side, and one residue is there in common in both of them, which is in between. So directly utilizing these two, you can keep one amino acid in common, and you can try to see whether the tripeptide made out of this combination will give you better result or not. Getting it or not? So two dipeptides, one is there and other one is this side. One is this side, other one is this side. One residue is common in between. So you can have a tripeptide, and you can see whether the tripeptide generated out of these two dipeptides gives you better score or not. So this is the tripeptide which is inserted into the active site, and what we got, GWE. One peptide was GW, other was WE. So we created GWE with the combination of these two, is giving better energy score than the previous ones. Then you add 20 amino acids to the N-terminal, you get 20 peptides, tetrapeptides, dock all these tetrapeptides. The one giving you the best score is the best tetrapeptide which is docked. Tetra ke baad mein you add 20 amino acids at the C terminal, one by one, you get 20 more peptides, dock them, you get the pentapeptide, right? And you can see here, as you keep on increasing the length from di to tri to tetra and to penta, your energy scores keep on increasing, right? Keeps on getting better and better. Dipeptide was 6.2 minus 6.2, tripeptide minus 6.8, tetrapeptides minus 7.6, and pentapeptides. And simple modification to the pentapeptide, we usually synthesize peptide acids, right? Amine at the N-terminal and carboxyl group at the C terminal. You can also use another resin which is called ring amide resin, where you get amide group at the C terminal. If you have amide group, then the score is even better than the acid. So these two were sent for activity, and one of them gave very good activity. To begin with, that is the best activity. You can further modify the structures and get optimized structures which give you better animation, right? So this is generated out of the one which is already there, utilizing in a published paper. So if it is published, that means it was of no use, right? Otherwise, the one who discovered that would have patented that and would have sent it for development. So that was not developed because that was not a good inhibitor. So these, if you simply optimize the structures by adding some of the groups, changing some of the groups, making this peptide a non-peptide, then you can improve upon the activity. Fine? Yes? No. There is one more example. I am not going to take it. We are already exceeding the time. Another good thing I wish to tell about these tripeptide structures which were docked in this structure. This is the best diagram that I have. The beautifully sitting peptide molecule, tripeptide, into the active site of the complete protein structure. This I am showing because I wish to communicate that if you synthesize using amino acids, you get better molecules to be utilized as drugs, right? Why? These are natural molecules. Okay? What should interact better with a protein having amino acids is the fragment made up of amino acids, right? So amino acids gives you more space filling model than the small molecules. Other thing is small molecules are rigid molecules. You get rigid structures. Here you get much more rotatable bonds which will be flexible, which will get docked in a better sense, which will combine and join into the active site in a better sense, and these can be degraded very fast as well. They work in nanomolar concentration, right? The only problem is you need to inject them, and there is no problem in injecting if it is saving the life. Then there is no problem in injecting also. They will be costlier than the small molecules, but when there is no molecule available, then these are the remedy which is there available safely, can be utilized for wherever there is a life-threatening situation. You can utilize these peptides. Another best thing about them is they can handle the mutations beautifully. I synthesized these molecules, I docked and synthesized these molecules. I have not revealed the sequence here. Is giving you minus 8.4, where the available drugs which are there, FDA approved drugs which are there in the market, you must have heard about pyrimethamine and cycloguanil. These are being utilized in the malaria, right? Is giving much better energy score than the available drugs, FDA approved drugs, these peptides, right? Then the mutated structures of the same strain. Quadruple mutant means 4 residues in the active site have mutated. Then nothing can bind, right? But these molecules are binding with the mutated strains. These are binding with a better affinity. The one which is binding with the natural protein is also binding with the similar or better affinity even with the mutated strain. So they can handle the resistance also. This is the beauty of designing peptide fragments against the target macromolecules, right? So this is the summary. The structure-based rational drug design approach saves time, effort and money rather than synthesizing thousands of molecules, wasting laboratory time. You can see what is binding, what is not binding, then start synthesizing. At least some rationale is there that this should bind, this should not bind, okay? And use of available chemical databases you can use, which have not been used for any activity. आप उसको use कीजिए, उसको आप you can even procure that molecule and see, because nobody has tested that for malaria, for coronavirus, for diabetes, for this and that. If it is hitting some of the pathogenic target, then you dock that and you see if it is giving activity, then take it out from the market. You need not synthesize that molecule, that is available. You can purchase that. Once it gives you very good activity, then you can start synthesizing in larger quantity for development, right? Drug repurposing strategy is being used every time. Whenever a new pathogen comes, all these database drugs are tested. Initial thing people do is this one, okay? Then retrieval of available compounds is possible the way I told you. Use of more focused medicinal chemistry tools to generate active analogs very fast, right? The process is more specific. So you know what is binding, what is not binding. You use the functional group or you do the modification according to the structure of the active site. And finally, it is fast screening method, high throughput screening methods. Multi-stage targeting is also possible. Same molecule tested on multiple targets is also possible. You can use that. This is the group. One girl which is sitting at the right has submitted the thesis in March, has got a job in Haryana Central University. This is the research associate, women scientist working in my lab. Other one is these two are the PhD students. He will be submitting thesis this year. Kanika, she will also submit the thesis this year. This was the research associate working till last year, has left, has got a job. And these two MSc students completed their dissertations last year and have left. New students will come this year. Thanking everyone for your patience and listening carefully. Any questions you should have are welcome. Though
So, this is pattern of pathogen mutation. You take inspiration from all the pathogen mutations. So that you can design a drug. Just take the pathogen and just take out the harmful part of it and use it.
I am not a molecular biologist, so what I can expect is some reverse method the pathogen might have adopted. So the translated protein is doing its function. If that is not able to perform the function properly, the reverse signal will go and the genetic material will modify such that a new protein is translated which is now able to perform the function. Right? We don't know what has happened. Molecular biologists can tell you in a better sense. I don't know why it is mutating when something is binding, because some information has gone in a retrospective way to the system and the genetic material has modified itself to generate the same protein with modified active site, right? And that modification will allow the substrate every time. Substrate is same, that is not modified, right? So it will allow the substrate, but it will not allow your molecule to bind. That information is very important, right? So the molecule which was working 20 years ago and 2, 3, 4 generations of the same molecule has come, right? A new one is binding, new modification has come to the active site. It is also possible the one which was working 20 years ago can work now, right? Because substrate is binding. So you do not know whether the molecule which has gone out from the market because of mutation that time, this new mutation may allow that molecule to bind now. That is also possible. So keep on trying those molecules also which have got resistant 20 years ago, right? Which were not binding that time may bind now with the new mutations. Getting it? Or, I hope you followed. Okay. I'm sorry, I forgot your name.
My name is Soul, and I'm from so people who born after 25, they will still
Maybe quinine may work on them, right? Because the mutations which have happened over there, the way I told, mutations which have happened over the years, right? And nobody has tested them once again, whether the one which was because of mutations, the thing which have got eradicated from the market, now nobody has tested, so that can be tested once again, whether the new pathogen, mutated pathogen is binding with that or not, right? There is some force field which calculates all type of interactions which are happening, which are not physical interactions, right? Supramolecular interactions which are utilized. One of the force field which is used in Autodoc, which is a free software, you must have heard about it, that utilize Amber force field, which calculates all the supramolecular interactions, but it doesn't calculate the charges of the atoms, partial charges every atom has, right? And another modified version where multiple docking is allowed, that is the Autodoc Vina, that allows another knowledge-based force field where the partial charges of atoms are also used for in the calculations, and also that accelerates the calculations, right? So that multiple results from the same software are possible. Autodoc you can dock only one, but for Autodoc Vina you can calculate multiple results in the same run. More questions? Nobody. I am available in 326. Anybody interested in getting further information about